with the voice. Mhm. Okay. Okay.
Yeah.
Mhm.
Thank you, Jennifer, for this very kind introduction. All right. Yes. Good. All right. Okay. You should be able to see my entire screen here. Okay. Good. Okay, so cool. Thanks for the introduction, and thanks Jenny, Judith, Dimitri, and Giovanni for the invitation. It's really a massive pleasure and an honor to speak here in the Myeloid network. As Jenny already introduced, my lab is in Milan, in Italy, the San Raffaele Telethon Institute for Gene Therapy, which is a pretty unique place. It's basically where some of the first gene therapies for genetic diseases and now cancer have been developed. So this was really one of the reasons why I decided to join this place, because at the time I joined the institute, I was thinking about a future in which we could manipulate and genetically engineer the immune cells and macrophages. And we are getting close to it. It's not really, we're not there yet, but my long-term vision at the time is actually proving to be sort of correct. So today I will mostly speak about key interest of the lab is the molecular and then how the environmental regulation of myeloid cells. And I will use mostly cancer as a paradigm. So very brief introduction, although it's for surely not needed for this audience. Macrophages, of course, are cells that are distributed basically everywhere in every organ in our body, where they play lots of non-immunological and some immunological functions. And most of these non-canonically immunological functions pertain to the maintenance of homeostasis of tissues. And actually, this wide complexity of physiological properties that macrophages can acquire and can develop are frequently co-opted in an aberrant organ like tumors. So in my view, tumor-associated macrophages or TAMs represent co-opted and aberrant version of physiological macrophages, where both the immunological and the non-immunological properties of these cells are co-opted to support disease. Now, understanding how these cells operate in disease and modulate the functionality of these cells, or engineer the functionality of these cells in cancer, require a very detailed molecular understanding of how the cells become and act, become who they are, and act the way they do. And this is where my previous life as a molecular genomic immunologist comes in. So in this slide, I'm summarizing really many years of research from many different groups. And this is sort of a genomic view of how we can rationalize the functionality of macrophages and actually of eukaryotic cells in tissues. So if we consider and if we assume that the functional properties of a cell are given, at least in part, by the set of genes which are expressed at any given moment in a cell, now the repertoire of enhancer is a main driver and regulator of the genes that get expressed in cells. Now, in turn, the activity of enhancers in the genome, which are thousands, hundreds of thousands possible enhancers, are regulated by a remarkably simple organization of transcription factors. So in particular, macrophages have been instrumental to define this paradigm, where identity signals like MCSF in macrophages stimulate the activity of transcription factors that are of the myeloid lineage. We call this lineage-determining transcription factor, and P1 is an example. Now, these transcription factors usually act as pioneers, meaning that they are able to access their binding site in the genome even when the chromatin is closed. And when they bind to these thousands of sites in the genome, they can make these sites accessible for other transcription factors. That's why they are called pioneers. And now, based on the activity of these pioneer lineage-determining transcription factors, then these sites become accessible to other types of transcription factors, including those that are activated selectively in specific tissues, like SPICE in the spleen or GATA6 in the peritoneum, and many other in different organs. And also, these sites can be accessible also by transcription factors like those of the NF-kappa B, STATs, and IRF family, which normally cannot bind DNA in a close conformation. So if we put together these three layers of regulation, we get basically an identity and a definition of a core set of a macrophage enhancer repertoire, which then is subsequently specified by the combinatorial activity of tissue-restricted and stimulus-activated transcription factors. Now, this was found in macrophages and was defined, these properties in these cells, but it's basically a very general property of eukaryotic cells. And more recently, my lab, and this I really have to acknowledge, a couple of friends that really were drivers of this area of research, and these are Leigwanger and Hidalgo, where we basically applied this same concept of genomic regulation based on enhancer control and microenvironmental response, also to neutrophils, which are another type of myeloid cells, which are short-lived, but are still able to undergo plastic and dynamic adaptations. And in particular, in this study here by the group of Andres Hidalgo, who was one of the first to show that in homeostasis, neutrophils can acquire organ-specific phenotype and transcriptomes. And then we also show how mostly in the blood and in the tumors, now different subset and different activation states of neutrophils can be observed based on the type of stimuli that these cells get exposed to. Now, to make things even more complicated and more dynamic, there is a concept here of that Jennifer alluded to, which is this concept of epigenetic memory or short-term memory, of innate immune memory or innate immune training. There's lots of different names. And this idea stems at the molecular level from a very simple observation that we did 10 years ago, actually, more than 10 years ago, when I was a postdoc. So in these experiments, we were studying at the dynamic regulation of enhancers in a primary differentiated macrophages, BMDMs. And here we were doing chip sequencing for this histone mark, H3K4 monomethylation, which is a mark that identifies enhancers genome-wide. This other mark here, histone acetylation, which is a proxy of transcriptional activity, and then binding of the transcription factor, myeloid lineage determining transcription factor PU1. So basically what we found in these experiments is that if we took macrophages untreated or stimulated with LPS for 4 hours or 24 hours, what we saw was a very dynamic control of enhancers. So in this example here, basically you can see that there's an enhancer that is pre-marked in unstimulated cells. And now what happens is that this enhancer is that this region acquires histone acetylation. So that's a poison enhancer that becomes active after stimulation. There were like opposite behaviors, so enhancers that were already marked by the monomethylation, which basically lose acetylation after stimulus, and these enhancers that get repressed in their activity. But what we were surprised to find was this class of enhancers that we call latent or de novo enhancers, as a group of Chris Glass showed basically together with us, is that we find those regions that basically lacked this key mark of enhancer, the monomethylation, and that acquired this mark de novo after cells were stimulated. So the key point here is that there's these regions in the genome that get functionalized de novo and that become available to all the other transcription factors. And then the cool thing about these latent enhancers is that not only they are generated de novo after stimulation, but when the stimulus terminates, these enhancers persist over time. And when macrophages get a second stimulation with the same LPS stimulus or even with a different stimulus, then these enhancers get activated much more fast or strong. So this was like a very initial definition of molecular mechanisms that would allow macrophages basically to adapt their behavior and therefore change the way in which the cells would respond to future stimulation. And I think this is very relevant now that we know how long-lived macrophages can be in tissues and how the accumulation of these de novo enhancers might actually control future responses of these cells to the environment. All right, so the other concept that is very important for this presentation is the concept of signal integration. Again, it's not really something super complicated, but this is very important for me to specify. That is, most of our knowledge about how macrophages and how cells in general behave is based on this model, which we call signal response. So we know very well how a naive macrophage that gets stimulated with different stimuli changes their transcriptome, proteome, cytokine, and phenotype profile. This is sort of spectrum view. But actually what we think is more relevant to study is how different stimuli that act at the same time, at the same place, in the same cell, get integrated into a functional outcome. And this is very important in the context of cancer, because of course their macrophages are exposed to a wide variety of stimuli. Some of those stimuli should be activating, some others should be immune suppressive. And therefore, it's not really these M1 or M2 definitions that make macrophages pro-inflammatory or anti-inflammatory, but it's actually the combination of these factors and how they elicit a specific and context-dependent response. All right, so in this context, the key focus of the lab is to understand the dynamics and the immunological microenvironment of pancreatic cancer. That's the type of disease we are mostly interested in. And of course, this is a tumor type with high with a very high medical need, unmet medical need because of the basically failure of conventional immunotherapies to treat this type of tumors. Now there's big excitement and progress about the possibility of using vaccination. But so we were interested in understanding how the microenvironment of pancreatic cancer is controlled. And in particular, because this type of disease is characterized by these two coexisting features. On the one hand, the pancreatic microenvironment very potently, so they don't really function in the microenvironment. But at the same time, and this is something that is not so realized, the PDAC microenvironment has also a high level of inflammation, which is linked to tissue repair, to fibrosis. And now I have highlighted here interferon, type 1 interferons in IL-1 and IL-1 beta as some of the key cytokines that reflect this differential behavior. Okay, so in this context, what we did a couple of years ago was to start and profile basically the human immune PDAC microenvironment by single cell sequencing. And what I'm showing here is a UMAP of tumor-associated macrophages or TAMs, which we basically cluster based on marker genes. So we could identify different clusters, most of which were already described. And we were interested in this one, which is marked here in red, that we refer to as IL-1 beta positive, because IL-1 beta, but also IL-1 alpha inflammasome, this is the gene encoding for COX-2 and other myeloid chemokines, were all marker genes of this subset of macrophages. Now, when we look at the transcriptional properties of these cells, these cells were very interesting. What we do here is a gene set enrichment analysis. So comparing the gene expression program of these IL-1 beta positive TAMs as compared to the remaining population of macrophages in the tumors. And then you see the gene ontologies that are upregulated in IL-1 beta positive TAMs include genes of the inflammatory response, angiogenesis, basically wound healing, and migration. So it's a canonical inflammatory response, I would say. But at the same time, the cells showed a depletion of programs related to interferon responses, antigen presentation. So suggesting that at the single cell level in the stem cells, these two programs, high inflammation and cytotoxic immunity, interferon responses, were differentially regulated and actually in an opposed manner. And of course, we could do these clinical correlations and show that when stratifying patients for the abundance of the IL-1 beta positive TAMs, that's the red line over here, we saw that patients with high levels of IL-1 beta TAM signature had a poorer survival actually, as compared to patients with low abundance of IL-1 beta positive macrophages. All right, so we went a little more deeper into this to understand what is the origin of these IL-1 beta positive macrophages. Are they monocyte derived, or do they reflect a local activation or differentiation of pancreatic macrophages in the tissue? So these are single cell sequencing of mouse pancreatic tumors. This is the KPC model. And we basically, what we do here is we cluster the myeloid cells from the blood and the tumor. And on the left part of the slide, the color reflects basically the clusters of macrophages that we identified. So these are in red. This is the mouse equivalent of these IL-1 beta positive TAMs. And here on the right part, the color reflects the tissue of origin. So yellow here is the blood, blue is the tumor, and there's some pancreas here in red, which we don't really see because it's behind the blue dots. And now in this case, the arrows are RNA velocity analysis predicting the differentiation potential of cells along these putative differentiation trajectories. So you can clearly see how there's a series of trajectories that basically converge on this population here of Al1 beta positive macrophages, strongly suggesting that these cells are monocyte derived. Now, I'm not going into the detail, but we actually could prove this in mouse models by using lineage tracing. So we now know, and this also has been shown by subsequent work from Florent Genoux and many others, and showing that Al1 beta positive macrophages, monocytes, basically, they sort of represent, they seem to represent the immediate progeny of monocytes that infiltrate tumors from the blood. Okay, so what is driving this transition? These blood monocytes, tumor macrophages. What is the signal that is driving this transition? So to do this, we basically analyze the genes that follow this trajectory, and then we applied a set of gene sets that were available in the lab and that we could associate to known factors in the microenvironment. And the strongest enrichment we found was for this class of genes, which are induced by prostaglandin E2. I will get in a moment to describe what PG2 is. And basically what you can see here is that the genes that are induced by prostaglandin E2 are highly and selectively enriched in this transition and driving the Al1 beta positive macrophages. And also, and this is of course known from many other studies, the levels of PG2 are elevated in human and mouse tumors. And there's really a massive body of literature showing how prostaglandin E2 is a fundamental regulator of the tumor microenvironment. I'm reflecting here, I'm highlighting here these studies from the group of Santiago Zelené and Caetano Reis-Souza, as these were some of the first studies showing that depletion of COX-2 can reprogram the immune microenvironment and promote tumor control by the immune system. Okay, so what is PG2? It's a fascinating molecule. It's basically produced, synthesized by any cell in the body, and the level of this molecule increased in any context of stress, infection, cancer, damage, or whatever. Now, mechanistically, PG2 acts in a pleiotropic manner, so there's lots of cells that express the receptor for PG2. If we focus on immunity at large, a very well-established function of prostaglandin E2 is to amplify inflammatory responses. So cells stimulated with PG2 basically tend to increase the production of chemokines and some inflammatory factors like IL-1 that favor the recruitment of immune cells. PG2 is also inducing vasodilation, edema, fever, so all signs of inflammatory responses, which are actually the signs that get inhibited when we use non-steroidal anti-inflammatory drugs. At the same time, prostaglandin E2 is able to potently suppress cytotoxic immunity. It basically reduces the activation of CD8 T cells, of NK cells. It limits induction of interferons. And then again, in stem cells or epithelial cells, really PG2 is able to activate stem cells to regenerate tissues. So basically in a single molecule, we are including basically some of the activities that one would want to have to restore homeostasis of tissues. You want to suppress cytotoxic immunity to avoid excessive immunological damage to the tissue. You want to activate an inflammatory response to favor recruitment of cells that would be protective, and at the same time, you want to stimulate tissue repair. So that's probably why PG2 is so important and in cancer. Okay, so how is PG2 contributing to the physiology of Al1 beta positive macrophages? In these experiments, we take macrophages or monocytes. We stimulate these cells with TNF-alpha, which is also enriched in the microenvironment of pancreatic cancer, alone or together with PG2. And then we measure Al1 beta. The results here are pretty striking to me. If you give macrophages TNF or PG2 alone, very limited Al1 beta gets produced and released. But if you combine these two stimuli together, then the macrophages became very, very potent and very able to produce very high levels of IL-1 beta. This is for both for macrophages and monocytes. Now it's not just IL-1 beta that behaves in this way. So in this heat map, we basically show here, we were basically seen here the genes that are poorly induced by PG2 alone, poorly induced by TNF alone, but are synergistically induced by the combination of PG2 and TNF. And you see that you get a very peculiar gene set, which contains IL-1 beta, contains CXCL1, COX-2, CXCL2. These are all markers of IL-1 beta positive macrophages. But also you get further upregulation of IL-10, IL-33. So it's a sort of, I would say, very specific inflammatory module that contains both cytokines that promote tissue repair and also chemokines like IL-10 that would suppress cytotoxic immunity. And this very same signature is selectively upregulated in IL-1 beta positive macrophages as compared to the monocytes, showing that the IL-1 beta positive TAMs acquire this program as they get into the tumor. All right, functional experiments. If we knock out COX-2 from tumor cells, and again, we are just repeating previous experiments in a different mouse model. That's the light blue line. So tumor cells that cannot produce PG2 anymore, they can basically engraft into immunocompetent mice, but then these cells are controlled in a T-cell dependent manner. And then how about IL-1? If we knock out the receptor for IL-1 in tumor cells, and that's again the blue line here, we basically mirror the phenotype of the COX-2 tumors, which means that IL-1 has to signal in the tumor cells in order for the tumor cells to grow in vivo. Now, as a control here, we're basically using this experimental condition where we take IL-1 receptor 1 knockout tumor cells, re-express the IL-1 receptor, and that's the pink line, and you see now how these cells basically grow back as the control. So the key model that we end up here is that in a growing tumor, the inflamed microenvironment of a pancreatic cancer where tumor cells and possibly other cells produce prostaglandin E2 and inflammatory factors like TNF, locally condition infiltrating monocytes into upregulating this IL-1 beta driven gene expression program, and then this IL-1 is fueling and is favoring the growth of tumors. But of course, no, this interaction means that there must be some local interplay. I'm gonna go pretty fast on this because this is basically a second part of the talk, which is mostly unpublished. So to start understanding how IL-1 beta is acting on the tumor cells and contributing to the growth of these tumors. So we first define this T1RS. It's a gene signature, tumor IL-1 response signature. It's basically a set of genes that is induced by IL-1 in tumor cells. And then we will look in human PDAC cells, tumor cells from PDAC patients. And we plot here the expression level of this T1RS, IL-1 response signature. And you see how there are some cells in the tumor, in patients, some tumor cells that express very high levels of this IL-1 response. And then when we compare these inflamed tumor cells with the non-inflamed tumor cells, and we do gene set enrichment analysis, what we found is that shown here in this plot is basically the gene program that are high in these inflamed tumor cells are of course inflammatory response, TNF signaling, but that's expected. We also see upregulation of programs like EMT, KRAS, TGF-beta, P53. So these are all programs that are associated to more aggressive tumor cells. So the conclusion here is that you get local IL-1 beta production by the tumor cells, and then IL-1 signaling in tumors is basically amplifying the aggressive capacity of tumors. I will skip this part here. I will just go very briefly to this analysis. In the paper, we use an initial preliminary type of spatial in situ RNA hybridization approach with a very limited panel of genes to show that in human pancreatic cancer, there's a spatial co-localization of Al1 beta positive macrophages shown here in red, and these are two different samples, and this subset of tumor cells expressing the Al1 response signature. That's the T1RS positive tumor cells. So we basically reinforce the model as before, but this time we include also the spatial co-localization. So the subset of Al1 beta positive TAMs co-localizes in proximity with a population of tumor cells, showing high level of inflammatory response genes and displaying high aggressive capacity, high potential for EMT, higher proliferation, and increased expression of oncogenic signaling. Okay, so now MyLab is really going forward in looking more into this interplay from clinical translation, mechanistically, how is this linked to the initiation of tumors, whether this pathway is actually relevant for tumor types other than the pancreas. And this is what is most exciting for us at the moment, is how spatially are these niches of Al1 beta positive macrophages organized. I will tell you later why I think these are niches. Okay, so I'm going to go very quick for all of this. Clinical translation, of course, no one here claims to treat PDAC patients with aspirin, although there is, epidemiological evidence. And again, here the group of Santiago Zelené has shown that inhibition of COX-2 can synergize with immune checkpoint blockade or chemotherapy. And trials are, I think, now on the way to understand whether COX-2 inhibitors or inhibitors of PG2 receptors can be used to as a combinatorial therapy together with conventional immunotherapies. And in terms of initiation, tumor initiation, of course, there's a long-standing epidemiological evidence that blocking COX activity is associated to a protection from the development of gastrointestinal tumors. And of course, the relevance of IL-1 in the initiation of tumors was shown in the CANTOS trial, where you can basically see that in patients that receive canakinumab, which is a blocking antibody against IL-1 beta, and then reanalyzing this data, one looks at the incidence of lung cancer. You see how, as compared to the placebo control, the patient group with highest dose of anti-IL-1 beta is showing a significant reduction in the incidence of lung cancer. And now this is becoming very, very relevant and very hot at the moment, because now we are, again, starting to reappreciate, how environment contributes to cancer initiation in a way that doesn't really depend on mutations alone. Again, I want to highlight this, a couple of papers by the group of Charles Swanton. In this one here, it's basically shown that in patients with lung cancer that do not smoke, exposure to the environment was found to trigger damage in the lung and infiltration of macrophages and production of IL-1 beta, which then would basically reactivate stem cell programs in mutated epithelial cells of the lung, and therefore leading to this increased incidence of tumors. And another very interesting paper that came out not so much ago, but there was many other, is this study by the group of Miriam Merad showing that in aging, alterations of the bone marrow and the increased inflammation in IL-1 beta and IL-1 alpha in this case, synthesis by myeloid progenitors, was associated to increased development of tumors in aged mice. So in general, there is this idea that IL-1 can contribute, or PG2 can contribute to the initiation of tumors. And in agreement with this notion, when we are start to look at how this PG2L1 pathway contributes to the initiation of tumors in pancreatic cancer, basically. So we started doing this by analyzing available data sets. So this is mouse models of pancreatic cancer, where we available single cell studies by the group of Scott Lowe and Dana Peer. So basically we could use single cell data from tumor and epithelial cells from the pancreas in these various mouse models. So we can get normal pancreas, mutated pancreas with no tumors, early lesions, and then established PDAC or liver metastasis. Now, when we look at the expression level of this, of our L1 response signature, you can see how this is elevated in tumor as compared to the normal pancreas. But actually, you can see how the highest level of inflammatory reprogramming are seen in these early lesions, both early and late PDAC. And even simple activation of KRAS with no over tumor is associated to this upregulation substantially of an inflammatory program. And this is also found in human when we look at early lesions as compared to tumors that are poorly differentiated. Not only that, if we look at patients with pancreatitis, and we know pancreatitis is a major risk factor for the development of pancreatic cancer, we can see L1 beta positive macrophages that are enriched and that basically infiltrate the pancreas of these type of patients. So what we are doing now is actually trying to study this in a mechanistic manner, establishing model for more reliable pancreatic cancer initiation and seeing whether inhibition of COX or L1 and can contribute, basically can delay the formation of tumors by possibly modulating the metaplasia that acinar and ductal cells go through during the development of tumors. We're also looking this in human samples. Again, these are really more ideas. We are doing this right now, but we have projects in the lab that are focusing on early lesions of pancreatic cancer, and in particular this IPMN lesion. IPMN stands for intrapapillary mucinous neoplasm. It's basically considered to be a precursor lesion of pancreatic cancer in some cases, not in a subset of cases. The advantage of studying this lesion as compared to panins is that these IPMNs are very, very frequent. So we are now using spatial transcriptomics to understand whether IPMN lesions that further develop into pancreatic cancer, and we can do this because we have matched and longitudinal sets of patients, will basically predict the tumorigenesis in humans. Okay, second, other part is, so we talk about the initiation of pancreatic cancer. How about the remaining tumors? First question is L1 beta positive macrophages just seen in pancreatic cancer, or are they a common occurrence in human tumors? So in this case, we do a bioinformatic analysis. We basically collect the dozens of studies doing single cell sequencing in human tumors. We annotate the macrophages, we analyze the cells in an integrated manner, and then we call the different clusters. And it's actually very, very interesting to see how in this UMAT that you can see here on the right part of the slide, the colors are they reflect the clusters of macrophages by putting together the entire set of TAMs from different 18 tumor types and more than 300 patients. So you see that L1 beta positive macrophages with the same L1, COX2, L1 gene programs can be detected in basically all types of tumors. And now here you see the different tumor types. The red is shows the abundance of L1 beta TAMs. So you can see how in each tumor you can get between 5% and 20% of the macrophages that could be annotated as alpha beta positive terms. So this is a general occurrence. What is the clinical correlation of this? It is really context specific. So there are some tumors like liver cancer, lung cancer, kidney, and GBM, where we can stratify patients, and we see that patients with high levels of alpha beta positive TAMs have poor survival as compared to patients with low levels of alpha beta positive TAMs. But there are some other tumors, and I want to make here the specific example of colorectal cancer of macrosatellite unstable. That's a very peculiar type of tumor that shows higher responsiveness to immune checkpoint blockade because of the capacity of these cells to undergo more DNA damage and possibly generation of neoantigens. Now, in this case, you see that patients with high levels of alpha beta positive TAMs have much better prognosis as compared to patients with low levels of alpha beta positive TAMs. So I think this is very interesting because it shows, and we kind of know, that alpha beta is also needed to activate T cells. So my idea here is that in tumors where you can get some T cell activation, then the high levels of AL1 provided by the alpha beta positive macrophages might be protective. While in tumor types like the ones I'm showing here, where the T cells do not work as well, then the alpha beta would basically favor the growth of tumors and potentially be suppressive in other ways. But this is to be tested. These are data I'm showing and discuss here for the, of course, for the sake of science. Now, the last part of the talk, very brief, but no, we are very excited about this. We are really interested in understanding better the interplay between alpha beta positive macrophages and cells in the human PDAC microenvironment. So for this, we are using spatial transcriptomics. We are using the MERSCOPE technology, but it doesn't really, it works very nicely, but it makes really no big difference as compared to other types of technologies, in my opinion. We work with a custom panel of now 1,000 genes that we defined in a curated manner to basically cover the entire diversity of TAM subsets, as well as the other immune populations in the microenvironment. And the cool thing, of course, as you might imagine here of this type of analysis, is that you can get the gene expression program of these cells, and then you can locate the exact physical placement of each of these cells in the 2D microenvironment of the tumor. Okay, I'm going to skip this part here. We basically use this spatial transcriptomics. We are stumbling into lots of these B cell aggregates, and this is something we are looking also at in the lab right now. But now to go back to the inflammation and the L1 part, in this, these are selected, but it's kind of representative section of human pancreatic cancer. So where you see that we have this in yellow, these are the cells that we annotate as tumor cells. The red is the fibroblast. And now you can see that there's something that seems like a core of the tumor and some invasive margin of the tumor. And now here in red, we are basically showing the expression level of this IL-1 response signature. So first thing we notice is that there is some expression here in the core of the tumor, but the highest expression level that we get is actually just outside or at the periphery of the tumor. And now this part here of this part here of the tumor that we basically find to be enriched in L1 beta program contains some clusters of tumor cells. Like in this case, we are basically coloring tumor cells in pink. And you see this nest of tumor cells, which express the L1 response signature. And then here you have the magnification. And you see how these tumor cells are really cuddled by these yellow cells, which are the L1 beta positive macrophages. But also how you can get in, and these are shown in red, population of fibroblasts that also express L1 inducible genes. We saw this in these specific areas, Now, please notice these areas of these classes of tumors are really outside of what we would define as the core of the tumor. So we think that this may reflect initial sites of tumor cell proliferation and further de-differentiation. Even if we look at like smaller aggregates of tumor cells, and that's again this part here. Now the violet, these are tumor cells. You can see them like in this nest. And again, you see how you get lots of these Al1 beta positive macrophages in yellow, and lots of these Al1 responsive fibroblasts in red. And in this case, we also have epithelial cells of the acinar and ductal lineage, which all express this Al1 response signature. So I want to go back to this part here now. So using this spatial gene expression data, what we did was basically to quantify in a statistical manner the co-localization of individual subset of cells. And now in this heat map is the colors are, the red is more frequent interactions, basically the columns and the row indicate the various cell subsets that we identified. So taking an unbiased manner, you can clearly see that cells tend to distribute into neighborhoods. So it's not like cells are all distributed as compared to each other. There's some of them. I will not really go into the details of what these aggregates mean. I will focus on this one here, which is shown here in its highlighted and enlarged. And you can see how in this neighborhood, basically what we find is the Al1 beta positive TAMs. We found Al1 beta responsive tumor cells. We find Al1 beta responsive fibroblasts. We find Al1 beta responsive epithelial cells. We find Al1 beta responsive endothelial cells. So this is why I'm calling these niches, because it's basically co-localization of cells that within the same local microenvironment, they all express response to Al1, which really supports the view that when these Al1 beta positive macrophages locally release the Al1, they condition all the neighboring cells. So the concept we are using now to rationalize this data is sort of this niche identity. So as compared to let's say a classical view of the tumor microenvironment, where cells, of course, have different origins, different lineages, and they express different genes because they are different cells, and they interact. Now, if we consider the niche as as a functional unit of a tumor, then what you get is that you basically have a local production of IL-1, in this case, or PG2, and this will basically impart a concordant and spatially coordinated gene program to multiple cells in the same area. So this is basically, I think, the definition of a niche, right? And this would, again, speculative, but this is something we are working now to formally prove. There will be a beneficial organization, beneficial consequences of this organization, because then you would have different cells with different identities that express same genes or coordinated genes. The behavior would be modular, so in case of tissue damage, and we are seeing this in models of tissue damage or other tumors, that these niches are basically reproduced almost exactly. So basically, it's the niche that gets elicited in different tumors rather than the individual cells. And then again, this is speculative, but I think it is relevant. This type of organization would also ensure robustness of the system. In this view here of the cell as a functional unit, if we take out any of these cells, then the specific activities of this cell would basically drop. And then, therefore, we expect to have a therapeutic effect. In this model, if we take away one of the cells, because the other one can compensate also functionally and molecularly, now this would explain why we are not being so successful in treating tumors, in treating patients, by selectively depleting one single cell or one single pathway. I think the local organization of cells in a niche whereby multiple cells express very similar genes and they respond to local environment is what makes tumors so hard to modulate. no? Because it's not enough to take away one component. You probably need to take away the entire niche. And this is something we're trying to do using gene engineering, gene editing, and building on some of the gene therapy expertise in our institute to actually locally deliver factors that would disrupt the niche. But again, there's no data on this. I'm showing mostly ideas, but I hope that's going to be stimulating for the discussion. Okay, so in conclusion, and this is my last slide, we are working a lot on this, and there's still a lot of questions that we want to address. In particular, the organization, composition, and functional properties of these niches of Al1 beta positive macrophages. We think that understanding how these are spatially organized would allow us to understand what is the actual function, how comes that Al1 and macrophages facilitate tumor initiation and progression. Can we use it as a prognostic tool, like to have a biomarker and discriminate and identify patients at risk of developing PDAC? Can we use this for therapeutic intervention? And of course, generally, what is the interplay between inflammation and Al1 and COX2? I think this is really a central pathway across most type of tumors. Okay, so I'm going to stop here with the acknowledgements. Most of the work has been done by Nicoletta Caronni together with Marco Geno and Federica Laterza. Other people in the lab, they're working on different aspects, on different cells in the microenvironment. We have also very cool studies. We have not dropped our old interest in molecular regulation of macrophage activity, but probably next time I will be able to speak at this wonderful audience. I might also tell about these stories. The funding agencies, you for the invitation and all of you for the attention, and we'll be super happy to take any question you have. Thank you.
I have some questions. It's beautiful work, very, very exciting. So